patients and their families are way more powerful than they realize.
Big time, absolutely.
So this was first shown for the Huntington's disease field, and you remember the ice bucket challenge in ALS?
Yeah.
So much of what's going on now and therapies are going to be coming out that came from that.
I'm John Foxe, Director of the Del Monte Institute for Neuroscience here in beautiful Rochester, and thank you for tuning in to another episode of Neuroscience Perspectives. Today, I'm thoroughly thrilled to be joined by distinguished neuroscientist Dr. Anne Hart. Her pioneering research established the first models of human neurodegenerative disease in C. elegans. She's credited with helping found the community of researchers who have since been studying these diseases in invertebrates, and her group works to understand why neurons degenerate in diseases like spinal muscular atrophy, amyotrophic lateral sclerosis, ALS, and frontotemporal dementia. Her lab has also worked to discover new neuropeptide genes in C. elegans to understand roles for notch signaling and to demonstrate conserved genes for sleep regulation. Dr. Hart is a professor and the Chair of Neuroscience at Brown University. And in addition to leading her own research efforts, she plays a central role in shaping neuroscience education and mentoring the next generation of scientists. She's very much involved in Brown's training programs and has been the recipient of many teaching and mentoring awards. Folks, make sure to like and subscribe wherever you get your podcasts. And thank you, Dr. Hart, Anne, for joining us here today on Neuroscience Perspectives. I really want to dive in. I'm fairly sure you're going to know what the first question is, because our viewership, will be interested to know why worms? Why worms, of all things,
Oh, yeah. No, why worms is a great question, which I get asked all the time. In fact, my family, when I first started doing science, was first, why bugs? Why worms? But the reason is simple. Animals on this planet are all really very much alike, right? And so they're all very related. And so if you want to have a cell, or if you want to have a neuron or a nervous system that works, that detects the world around you and responds to it appropriately, the basics of how cells work is pretty much the same.
Right.
So if you want to ask questions about how neurons work and what happens when they go wrong, sometimes it's easiest to ask those basic questions in a very small animal,
right,
like the fly or C. elegans, little worms. They're the same size, I know, as like the period in a sentence. That's how big they are. They're tiny. But
less than a millimeter.
Less than a millimeter, yeah.
Amazing,
but they have 300 neurons, and they use those to navigate the world and respond.
And that's a key piece, right? Because those 300 neurons, that entire circuit has been mapped out. We know what connects to what, where it does it.
Yeah, definitely. One of the features of C. elegans is every neuron has its own name. We know exactly where it is in the animal, and we know which neurons are connected to which neurons.
Yeah.
So it's this is an advantage of the system, is we know exactly what they're working. I can go to each animal and say, that neuron right there does this, that neuron right there does that.
Yeah.
Yeah.
And I mean, it probably matters that it's kind of see-through as well, right? You can actually see into it.
Yeah. It's transparent. So we can actually, I can actually put the animals on a microscope and look inside the animal and say, that neuron right there. I look straight into the animal without manipulating. And then I can take it off the slide, put it back where it lives in little Petri dishes in the lab. It'll crawl away and do things. These are tough little animals that are easy to work with.
Right. And they breed fast as well, too, so you can do genetic manipulations.
Yeah, there are millions of animals in my lab at any given time. Millions. And because there are a bunch of them in, imagine you have a couple refrigerators, although they're kept warmer than that, from your kitchen. Something that size will hold 500,000 worms for us to work on. And so not only are they small and super inexpensive, right? But we can freeze them down, and then you can thaw them and bring them back to life.
They're super resilient.
Super tough. And so I have worms from when I first started working with them that I can bring back, quote unquote, to life, and then start working with them again. So They're easy to work with, and so that's one of the distinct advantages.
I love it, love it. And yet, as 300 neurons in this tiny little package, see-through, transparent package, is capable of pretty complex behaviors, right?
Yes. So the behaviors are, sometimes I say what we are limited by is our ability to figure things out. So we have learned over the years that they can learn, they can adapt to their environment, they can detect and avoid and remember dangerous things and things they like. Part of my lab works on sleep in C. elegans, so C. elegans also sleep.
Yeah.
And the same basic way cells change their function
Right.
when we go to sleep, and the same pathways that say it's time to go to sleep, many of them are conserved all the way down to
So
C.
there's
elegans.
circadian rhythms of a...
I'm sorry. That is the one thing they don't have.
Okay.
Because if you were a little worm living in rotting fruit or the soil,
Right.
you don't really care if it's light or
Light's
dark.
not a big deal.
But if I keep them awake when they want to sleep, they'll crash and sleep harder, right?
So they
and
need
they
their
don't
sleep.
do as well. Yeah, and they don't do as well if they don't sleep. So, and they sleep a lot more when they're growing
Yeah.
than they do when they're adults.
That's
which,
not-
if you've
yeah.
ever had kids, you know that's
Exactly.
a thing. Kids sleep a lot.
Yeah.
So, and not only is these overall things we're talking about conserved, but the same molecules are also involved. So if we try to figure out what's happening and how the animals control, okay, I'm going to sleep now, or we all need to get together and be quiet as neurons so we can sleep, those pathways are conserved across species.
That's outstanding, yeah.
Yeah.
So when somebody comes to you and say, are you really seriously telling me that this little tiny thing has something meaningful to say about the human condition, you're ready to go?
I'm ready to go. I mean, I will say there are things we cannot study in C. elegans. Humans have very complex behaviors. So there's some things we have to study in mammals or something. But if you want to talk to me, if we want to talk about cells and neurons and circuits and the very fundamentals, a lot of that stuff we can do in worms.
Right. So the channels in the synapses, the genes that code those, there's a lot of that that's conserved. I mean, this is an ancient species, right?
Yeah. This is both an ancient species, and I think they are more evolved than we are. So if you think about it that way, they have a generation. An animal is born, and within 3 days, it can have babies too. And so they have been through many generations of evolution, figuring out their niche, and they're really streamlined and efficient.
Fantastic.
We have not had that many generations compared to worms.
That's a good point.
We are bigger and more complicated, and there's much more going on up here.
Yeah.
But you're right, the fundamentals are just the same. And that's because many of the molecules are the same. And so when I want to model diseases, for example, sometimes I can look at a human protein and say, okay, this is the sequence, this is all the little amino acids that make it up, and I can match the worm protein up right next to it, and it's almost identical. And I can say, oh, if this change causes disease in humans, let's put that change into worms and see what happens.
Right.
Yeah.
When you first started out trying to do the worms, did you meet resistance?
Yeah.
Yeah, tell us about that.
This is common in the sense of if you want to start something new, there is an established way of people thinking about problems and how we can tackle them. And then someone comes up with a new technique or a new approach or a new strategy. And understandably, you have to convince everyone else that this is a good idea.
Right, whatever they've been doing for the last 20 or 30 years is...
Yeah, right. Yes, it's still good.
Yeah.
But this would be useful. What I'm saying now could apply to a new microscopy technique or the wild concept of we can put disease mutations in worms and learn about them. When you start out as a professor here in the United States, You are part of a department of scientists at a university, a research institute or a school, and you have a chair,
Yeah.
which I am now,
Yeah.
who runs the department. I remember my chair came in and said, "This seems like a real gamble, this putting diseases into worms. Maybe you shouldn't do that,
Yeah,
or
yeah.
you shouldn't rely on that for your career." I ignored him.
You ignored him.
I
Good,
ignored him.
yeah.
I just kept going. So we put, the first one we did was we took the change that causes Huntington's disease, and we put that into worms, and the worm neurons died.
The worm neurons died. Did
Yeah.
they develop a phenotype, like a behavior that seemed Huntington-like?
So this is both the strengths and the weaknesses of the worms. When we put the Huntington's disease is caused by, we have a part of our Huntington protein that has the amino acid glutamine, glutamine. And in patients, that's much longer,
Yeah,
so
right.
many Qs, and that's a problem.
Yeah.
So we put very long stretch into the worms, and it caused neurons to die only in old worms, which made sense in
Right.
a Huntington's
Yeah,
field
yeah.
because Huntington's patients don't have symptoms
Till
until
later.
they get older.
Yeah, yeah.
And so the neurons die, but worms can't show Huntington's chorea.
Right.
They don't
Like
they
the
don't walk,
movements,
they don't
yeah.
try and control limbs.
Yeah.
But for me, it was sufficient to say only expanded these repeats, only the big disease-causing repeats make the neurons die in the worms, and they die in older animals. And when we were able
So
to...
it has the same temporal
has the same
characteristics that you see in the...
lifetime, temporal
Yeah.
characteristics, the same length of dependence. And while we were working on that, we were also able to test things that help the neurons in Huntington's disease models, and those also worked in the worms. So
Right.
it was...
And that's a big piece of it too, right? If you have disease in a dish in a worm model,
Yeah.
you can test anything against that, right? They're cheap.
Yeah.
There's millions of them, and you could take a swing for the fences on any FDA-approved compound. Is that part of it, or?
That is part of it. I will say that C. elegans is not used right now.
I see, okay.
As much for drug screening, although I'm working on another disease gene now that causes a very rare disease called alternating hemiplegia of childhood. These patients are rare, but the
Right.
disease gene is in patients, and the worm gene are very similar. So we can put the patient alleles in by just making a little change with
Yeah.
CRISPR. And we're talking now about how we can do drug screening in those.
Fantastic.
What the worm model, people working in worm models usually do is try and use genetics. And so I will say that genetics, when I was in high school and college, seemed really painful and complicated and recombination, but as a tool to figure things out when we're working with, mice or
Yeah.
flies or worms, it's great.
Yeah,
So
yeah.
we try to figure out how the neurons die and how we can stop them by making a specific gene broken, which means the protein doesn't work, and seeing if that helps. Sort of the way you can think of it as analogous to you take a drug, put it on the protein, change how it works, and that helps the neurons.
Gotcha. Yeah,
So
yeah.
instead of going for chemicals, we're going directly to the genes and the proteins.
A thought that people might have is you've got worms, flies,
Yeah.
you get invertebrates, and then you move up to small rodents,
Yeah.
and we move on up into other primates and so on. And that the closer we get to the larger, let's
Yeah.
call it larger creature that's closer to the human
Yeah.
phylogeny, that you're getting closer to the conditions of a human. Do you buy that, or is there advantage or
Good.
disadvantage?
Good question. I mean, I think each of those models that you're talking about, when we're talking about disease, let's just
Yeah,
talk about
yeah.
disease for a moment, has its advantages. So I can have 1 million worms in my lab. I can do whatever I want to them. I can do experiments in 3 days.
Yeah, yeah.
And so I can try this idea, fail, this idea, fail, this idea, fail really fast.
Right.
And the same applies to our friends who work with fruit flies,
Yeah,
little Drosophila. I think a great way to approach this is try a lot of things in little bugs if you can. And when you think you have ideas, then I go to my collaborators who work with mouse models of disease and say, try this, or try this, or try this. And then if it works there, then we'll move to something bigger.
That's great. Yeah. So you are really the high throughput system at the base of the pyramid.
Yeah, we're both the high throughput system, and there are approaches we can use that would be ridiculously expensive in a mouse. Do you want to have 1 million mice to test one idea for one disease?
Right.
We can't afford that as a society.
Exactly. Yeah. Very good. That's really amazing. You were talking about that chair of yours. We won't call him out, right?
Oh.
but
I should say, brilliant scientist.
Yeah, and I'm sure supportive in other ways. Yeah. What were the big hurdles?
Oh, well, we could talk about Thomas Kuhn and the idea about the scientific revolution and how ideas propagate through science. That's sort of like history and philosophy of science.
Yeah.
But
take us there, if you will.
Well, sure. What happens is we have a dominant paradigm, like DNA, RNA, protein, or, humans have complicated disease and little worms don't. And then we learn or try something new, and everyone's like, but this is the paradigm.
Yeah.
You have to really prove that you have something different. So when little RNAs came along that changed how other RNAs work, people and people had to really prove that was what was happening. In our case, we had to prove that there were enough similarities between the worms and the humans, and we were finding useful things out. Because,
Yeah. And so you were pushing hard against
to change our
paradigm shift. Yeah.
We're pushing for a paradigm shift was what we were doing, right.
Structural hurdles in the academy as just another domain.
Structural hurdles in part would be funding.
Right.
Science is both not cheap and cheap at the same time, basic science.
Yeah.
Basic science looks expensive. If you look at it and say, millions of dollars on little worms. But if you compare that to the cost of trying to do the same experiments in mice, oh, wow, that's a real bargain. We should do that.
Yeah.
And also, so one of the hurdles is when trying something new, you need to find someone who will fund it.
Yeah.
And so...
And I think sometimes that equation too, people say, oh, you're spending millions of dollars on worms, but you're trying to solve neurodegenerative diseases, and on the back end, the cost to society is in the hundreds of billions. And so we're spending, we're really nowhere near investing enough in this front end, because if we solve this, then there's hundreds of billions in savings, not to mention relief of human suffering.
Right, but it's the paradigm shift or the concept shift had to be that we can learn something useful from the little worms. Right.
I'm fascinated. So we've gotten off on this, but let's stick with it because I'm fascinated. I want to know a lot about your transition into leadership roles and the maintenance of a lab. But let's come back to that because on this business of the funding and that, given that you're a leader at one of the great institutions on the planet, when you look at the current system, if you had the magic wand, are there things you would do to change it? Or has that evolved to be relatively close to the best paradigm?
I mean, some things, for example, I can mostly speak to what the United States and the NIH and the NSF is doing. Some things they've done are good. There are now more awards for people doing innovative stuff.
Right.
Compared to what there used to be. But there's still one of the things that we do really well, at least historically in the United States, is have other scientists read your proposal, critique it, and decide these are the good proposals, these are the bad, not as good proposals, try again.
Yeah.
And this iterative and peer review has been a major reason we ended up funding things that worked.
Yeah.
And it ended up propelling, one of the reasons that the United States has been historically such a leader
Yeah.
in all those Nobel Prizes, all of these breakthrough drugs, all of the pharmacological innovations we can do.
Yeah.
But so I think that unbiased peer review and the push to put in more innovation awards has been helpful.
Just to back up on that, in some ways, right then, there's a push through the peer review system to get the things that are almost baked into work. Is the system a little bit inherently conservative then in a domain where we shouldn't be?
As I said that, I thought you will notice that tension. Historically, the tension was they wanted things that would work by peer review and things you recognized, which is why I think it's been really helpful for the NIH to put in innovation sorts of awards.
Right.
And the NIH always
has space for
some
people.
high risk, high gain.
Right.
And when you write one of those, I actually write in this. This is high risk because is the worm really a model for this? And I said, but this is high gain if I succeed because $1 billion problem in dementia, for example, or there is no cure for this rare childhood disease. And if we succeed, the payoff is
massive.
Big.
And so infusing more of this kind of innovation, swing for the fences kind of mechanisms in there, that would be one thing.
I think that would be good. More of that. And I will say, and this is actually something that is very unique to the neurodegenerative field, is the power of patients and their families to push both the NIH, whatever government you're in, and to help researchers do researchers is patients and their families are way more powerful than they realize.
Big time, absolutely.
So this was first shown for the Huntington's disease field, and you remember the ice bucket challenge in ALS?
Yeah.
So much of what's going on now and therapies are going to be coming out that came from that.
Yeah.
So, and
there's nothing more compelling than the mother of a child with neurodevelopmental disabilities. These people tend to be, their communications, because it comes straight out of the core of their being, right? So patients and their families are also much more savvy now. When they start working together, you can look at examples of where patients and their families have gotten together and said, and worked with scientists who said, this is our best approach for going at something. Yeah.
And
this happens in my own field
it.
because I work in rare disease and neurodevelopment, and those patient groups that get together, apart from the fact that they provide extraordinary support for each other, and they're a base of knowledge themselves of the things that worked with their kids who have a very specific clinical disease. They are such amazing advocates, and they gather an energy around them that really changes the equation.
Right.
I'm just thinking more a little bit this business of the somewhat conservative thing that happens with government funding. We need to make sure that the American taxpayers' dollar is spent on things that are going to work. Right?
Absolutely. Well, there's tension in your sentence. We need to spend American tax dollars on things that we know are going to work. We don't always know what's going to work, which is why it's almost like we need to place educated bets so that we have both high risk, high gain, in addition to we think this will probably work.
Yeah. But it brings me to the role of philanthropy in this, because here we would say, that's where the monies that come in from philanthropy allow us to take risks and to write projects that are out there, blue skies kinds of stuff that we wouldn't be able to try with government money.
Right. I think every one of the disease projects I've worked on in my lab, definitely Huntington's disease, ALS, and now alternating hemiplegia of childhood, all of those were seeded by philanthropy from families and their foundations.
Right.
And all of those went on to get funding from the federal government after we had the information and the data and the results to say, look, it works. And
Yeah.
then we go to the NIH and they give you enough to actually, for example, push to figure out what's going wrong and how to stop it.
Right. That's so beautifully articulated, because I often have, people say, well, what's my few dollars? What difference is it going to make? And I say, you've no idea. Your dollars are different than the federal dollars because they allow us to do things that are just much more creative.
Yeah.
I love it. Yeah. Let's get into are you an accidental tourist into leadership, or was it always on your mind that you wanted to run a department and to paint on a broader canvas?
You're going to laugh. When first said, well, maybe you should think about being chair, I say, no, I'd rather jump out the window. Yeah. But there are different types of leaders. And I will say this often to young scientists I talk to now, is like, at the moment, being in charge of a department or being in charge of a big research program looks like that's impossible. That couldn't be for me. But actually, as we go along through our careers, first you're learning how to organize your own science project. Then you're learning how to organize the people who work with you in a lab. And then you're learning to work with other scientists in bigger collaborative affairs. And by the time you get around, in my case at least, to saying, could you be chair of a department of the 20, 30 people who you know best as other scientists, like at Brown?
Yeah.
It's like, yeah, I can try to do that.
Yeah,
I could do that.
If I may say, your leadership skills are totally evident right here. I think one of the big drivers is one's own personal passion, and it's oozing right out of you. You just can tell that this is what keeps you going.
Well, part of why we go into science, I mean, you know this, scientists and professors at universities, researchers at institutes, they could do something else.
Yeah.
They don't, if you don't stay in this game, usually, if you don't want to both figure out scientific problems and help people, but also teach, mentor, and educate other people. That's part of some of both of those parts of the equation are why many of us stay in academia or research institutes.
I like the way you also went from you start out with the small group and that, and then the next step before leadership as a chair was collaboration and community. Tell us about that. What do you think about, where does that role of collaboration and getting out of your own lane and out of your own box play for you?
When you say community and then you say getting out of your own box, I think of two very different things.
Okay.
When I think of community, actually, I sort of think of this as I was mentored and part of scientific communities that tried to help people, tried to do science better, tried to work together. I mean, all of us here stand on the shoulders of the giants before us who trained us or who mentored us along the way.
Yeah.
So you almost look back and you say, oh my gosh, that was so useful. What can I do to pass it on, right? Mm-hmm. And then from watching them, that gives you at least the initial skills to learn how to work with other scientists in larger collaborations.
Right.
So it's sort of a natural outgrowth of you learn to collaborate in small research groups even.
Yeah.
So.
You're the chair of neuroscience, and we live in a university system that's sort of, it's a couple of 100 years old in the sense of departments, right? There's a department of anatomy would be, and biology and chemistry because they were different disciplines. But science is not like that these days. And the departmental structure sometimes, grates with the way science is done. How do you deal with that in your leadership role?
That's a really good question. Science is actually interdisciplinary.
More than ever now, right?
More than ever, yeah. My lab, for example, could just as easily be in a genetics or molecular project. It's more a matter of who you want to collaborate with, what the sort of things you want to teach.
Yeah.
You could organize people in very different ways.
Right.
But if the groups get too large it's very hard for them to have the collaborative, supportive
Right.
interactions that are critical, I think, for educating and doing good science.
Yeah.
So we have to find some sort of smaller groups to work in. We'll be as, maybe thematic is pretty good choice.
Right. What do you do when you're stressed? How do you deal with that? You're never stressed.
Everybody needs something to do that isn't always on focus. I mean, humans need some relief.
Yeah.
So I read. When I was younger, we used to do a lot of hiking and backpacking, played amateur ice hockey. Now we live in Rhode Island right now. So we sail because Rhode Island is more water than land.
Yeah.
I cook. I like cooking a lot.
Cool. Yeah. So some physical activity and
physical
reading.
activity and reading.
Yeah. When you're reading, what are you reading? Do you step away from science?
Oh yeah.
Good.
For me, reading is escape. I mean, I was intimidated by my colleagues who were like, and then I read this very erudite book, and I'm like, that's good, but I would open up science fiction over here.
Gotcha,
and it's fantasy. So really,
a little
reading
escapism.
is my escapism.
Yeah, good. Yeah.
So because if your mind pulls away from the problem you're working on to do something else, whether it's cooking or hanging out with your friends or going for a hike, and think about something else, or go to sleep, you come back refreshed and you're like, oh wait, these two things go together. Now I could tackle it that way.
Very good.
So everybody needs a break.
I have a maxim for myself, which is if I don't have space in my life for a good book, there's something out of balance with my life.
Yeah.
And so I think it's really, really great way to exercise your mind in a different way outside of the academy. Let me see. I wanted to ask you a little bit. You actually mentioned a lot about mentorship. Mm-hmm. Young up-and-coming faculty at Brown University, getting their first graduate students. Do you have pearls of wisdom for them?
Work on important science, but be tactical.
Yeah.
Work on stuff you care about.
Right.
But, we talked about, you have to find a way to get yourself funded.
Yeah.
You have to work on a problem that you can make progress on. And you want to collaborate and be the kind of person people want to collaborate with.
Yeah.
So that's some of my advice. My other advice, and I bet you'd say the same thing, is find good people to work with in your lab.
Yeah.
And
lab
treat
chemistry and culture.
Treat them well.
Yeah.
Right. Yeah. You want to
tell
have
me
a
about treating them
cohesive
well.
team.
Yeah.
Oh.
Do you do extracurricular activities with the lab? Is that a piece of it, or
every lab is different. I don't actually have a lot of extracurricular activities with the lab. We definitely do stuff. We go ice skating sometimes, and we go kayaking sometimes, and my lab gets together sometimes and like crocheted little worms, sort of, and celebrate things. I was thinking more of somehow you want to do two things: is instill a team feeling in your group of we are in this together, we're helping together.
Does it get over into the world of almost like a family?
Yes, sort of.
Right.
But sort of they're like my academic children.
Yeah.
Right.
Yeah.
But also, and this you'll probably recognize this too, being a leader also means saying to someone sometimes, you need to do this.
Hard truths.
Hard truths as well.
Yeah. I read that I think you and I suffer from the same rare disease. You're looking worried. I call mine pathological optimism. You call it relentless optimism.
Oh, yeah, relentless optimism, yeah.
How does that play out for you? And why is that important?
There's a lot of, I tried that and it failed. Or I dropped, this mistake, this was wrong. This is an error. We're constantly, science, they tell you, put together a hypothesis and test it. And then most of the time it's wrong.
Yeah.
So being optimistic is useful.
Yeah.
And also figuring out the kind of stuff in lab you find joy in. So some of the stuff is small. I really like, making, the hands-on part of science I actually like. I don't do it very much anymore.
Right.
But I also like the part about teaching people to do the hands-on stuff. So in our field, we often have like lab courses.
Yeah.
Whether they are bringing people from all over the planet together to learn stuff, like in summer courses at Woods Hole Marine Biological Labs, or lab courses for grad students at Brown. I really like running and teaching those classes.
Yeah.
So find things that you enjoy doing that are also beneficial to your team and your science.
Yeah. Well, I think this goes to your leadership piece because people are looking, they need an optimist to get them through the dark times and the bleak times, somebody who sees a way out, and that's important. Last thing I want to ask you about, because I think our viewers really like to know when you caught the science bug. What was the 10-year-old Anne Hart like? And
that's interesting.
And your background, like, are you from an academic family or?
Oh no, I'm not. Yeah, so my family is all in business.
Okay.
And they're learning. And me learning. I really did no idea what I was getting into when I said I want to be a scientist.
Yeah. But when did you say that?
I don't know. In the sense of when I would say, you said 10-year-old. So I was given a terrible little microscope that was about this big when I was in probably second or third grade. And I definitely remember putting things on little slides and looking at them, and it's like, oh my gosh, a feather is really wild when you look at it and see, or the skin of a mushroom and things there. But I also like nature. I mean, I like bird watching. That's one of the things I do.
Right, yeah.
I'm not good at it. But so I always like nature and watching things.
Yeah.
But I also like, trying to figure out how things work.
Yeah. And what did your mom and dad say then, business people, when you said, I want to be a scientist?
Well, I think they have a hard time understanding that science is very much a apprentice, journeyman, sort of then professor sort of thing. It's like you go along and it's like, they would say things like, do you have a real job yet? And I was like, yes. I mean, but no. And then I would became a professor, and it's like, so this is a real job. I was like, yes, unless I don't get tenure, in which case I have to go somewhere. So finally I explained it to my business family as, what I do now is I run a small business whose output is educated people and science. And I have to do fundraising all the time, though.
Yeah.
Almost venture funding,
capital,
persuading people to put money in.
I love it. Yeah.
And that makes more sense to them about what I'm trying to do. I mean, when I started in science, I think my parents thought I would have to pay to do my PhD. I'd have to pay tuition like you do medical school.
Right,
but
that still
usually
astounds
not
people.
true. Astounds people. But that's because I think people also don't understand scientists in biology are not just sitting around reading papers, thinking and writing. People in the lab work 40, 60 hours a week at the bench, really hard trying to do their experiments.
Right,
so these are very committed young scientists who are learning how to do science, but also it's hard work.
And it's an absolute pleasure to have you in Rochester. Thanks so much for taking time out to be here with us on Neuroscience Perspectives.
It's been a real pleasure to talk.
Excellent.
Thank you.